you can all see, I'm going to go ahead and start. And what I'm going to speak about is a little bit of a change in tides from what we had heard earlier this morning. And I'm going to talk a little bit about chemical senses. So chemical senses, in this case, taste and smell, can be used to study neural circuits and information processing in health, helping to better understand basic neural mechanisms and how such mechanisms may be altered in different states of disease. Preclinical work in rodents and non-human primates may bridge the gap in understanding brain disease and in translating knowledge to sustain healthy brain function in humans. With a model sensory system and organism in place, single cell population activity and systems level correlation of neural activity were investigated in brain regions responsible for processing chemosensory inputs. So you can see I'm hovering my mouse over an icon on the lower right-hand side. This is a coronal section of the human brain where different internal structures have been highlighted. And some of the structures I'm going to talk about today include the striatum. You can see the dorsal striatum here hovering over in the green. I'm going to speak a little bit also about the insular cortex, and I'm highlighting that here. And that's also an area that's a bit internal, and it's a primary area that's responsible for taste processing, and it also plays a role in the processing of olfaction. So one of the first experiments I'm going to discuss with you is using magnetic resonance imaging, MRI, and this was using a ultra high field magnet. So it's a 7 Tesla strength. And we used non-human primate model, the macaque monkey. And you can see in this coronal image, this is basically the result we would obtain once we put the monkey in the scanner and we start imaging the sequence. And what we had done here was we did a functional imaging sequence to look at changes in the underlying neural activity over time. And the areas that we were interested in included those that we knew were responsible for processing taste. And what we could do is use atlases that were parcellated and defined on model or template brain images for this model organism. So you can see here through the axial or the horizontal plane, different structures in the atlas that we looked at, highlighted and contoured in green and blue. And you could see that this is what the image would look like if we were to scan the anatomy of the animal. So by doing that, we put forth a paradigm to go ahead and look at the activity as we issued different tastes. So in this case, we had sweet, sour, and salt tastings that we delivered to the mouth of the animal for 7 seconds. This animal was in an anesthetized preparation where the tastes come automatically to the mouth and it's rinsed out and flushed. And so the rinse that we use, you can see occurs for another 7 seconds and whereby we have a rest period. And so if we want to look at then the correlated activity in the brain, also called the BOLD activity, it's blood oxygen level dependent signals, what we could see is highlights of the primary cortical representation for taste. And that you can see is highlighted here in yellow bilaterally on the left and right hemisphere, the activation of the insular cortex. Now, the insular cortex can be subdivided based on its cytoarchitecture. We have the dysgranular insula, where we have granularity that is not in fine patterns, rather it could be a mix. And we have the mid-dorsal insula. So that's which is sitting on the dorsal surface of the insula, if you can follow my mouse here. And that is in a granular, a fine cellular layered organization. And if we move a little bit further anteriorly, we have the ventral insular cortex, which is agranular. It doesn't have as much fine granular cellular organization. And throughout the insular cortex, we can see that there are selective regions localized for the taste. And now this isn't a passive taste setting where there's no task engagement. It's just how the brain is processing these stimuli when we give it to the animal. And now to complement fMRI and get a better idea of what might be occurring at the actual cellular level, we went in with a 4 electrode array tungstens here. So it's a tungsten metal material. And we went ahead and we placed our electrode targeting the insular cortex. And we used the coordinates based off of fMRI to help us. And what you can see here highlighted in pink were the actual niches that responded to the taste, when we went in to record the multi-unit and population level activity. Now, what's interesting about the insular cortex is that within these cytoarchitectural boundaries, we can see that different activations may be selectively influenced by the ongoing physiological state, which includes satiety, anticipation, expectation, and the state of stimulus processing. And this is a signature that we're seeing across species, and that includes humans, macaque monkeys, and rodents. So I'm going to switch keys now to our other model organism, and that is the mouse. So what we have here in the schema is we have two mice interacting during a social interaction. And what you could use is a software, for example, Deep Lab Cut, to computerize the interactions and study them offline. And what we'd placed in these boxes were different types of animals that had their own social odors, that had their own individual odors. And what we were particularly curious about was how the odor relays through the primary olfactory system. And to examine the circuitry, we complemented that with what we might be able to measure from outside. And that includes, for example, pupil dilation to measure arousal, and sniff response to measure potential influences of valence or appetitiveness. And the methodology that we want to use to uncover what's happening in the brain includes fMRI and electrophysiology. For the sake of time, I'm going to focus here on from the fMRI aspects. So a good question is, how do social stimuli and social experiences influence odor processing? So we did this first experiment to help distinguish odor types in a state of passive odor processing. And here's the image of our MRI scanner. It's a 9.4 Tesla, so it's a strong magnet. And the animals that we're measuring go in. And what we do is we have these 3 animals placed in those social odor boxes to deliver their odors to the animal in the scanner. And we have non-social odors. So that includes a floral yang odor and the odor of peanut butter. So what we can do by fMRI is an odor stimulus classification. And we can take that in hand with the physiological readouts that we also applied. So for example, looking at the sniff responses, you could see here 3 odor trials collected during one recording. And we have the sniff response for peanut butter, top, yang, the floral in the middle, and the juvenile, an appetitive social stimulus. And what you can see is you either have an amplitude in the sniff response cycle, or you have an increased frequency, indicating what could be a level of increased appetitiveness. For the yang, however, you could see that there's a transient termination in the sniffing, which could indicate that it may not be as appetitive as the peanut butter or the juvenile. So looking at the bold activity, or that by fMRI, and doing a contrast, taking that activity correlated with peanut butter and that correlated with the floral yang odor, what we could see is that appetitive social odors drive the bold fMRI activity in the areas that include the anterior piriform cortex, which I'm highlighting over with my mouse, the olfactory tubercle, which is situated ventral. And if you go anterior, you have what's called the anterior olfactory nucleus and the olfactory bulb. So you can see this distinction when you start to do the classifications by different odor types. And now we wanted to know, what about if we modulated the social experience by evoking oxytocin release? So oxytocin is a neuropeptide. In the social realm, it affects perception, it can alter behavior, and also influence bodily physiology, such as anxiety levels. Oxytocin administered during social interaction with the same species, conspecific, resulted in increased exploration events. This is found by Otto et al. In the lab of Wolfgang Kelsch, where I'm also conducting my postdoctoral research. So what you could see is during a sample phase where 2 mice interact, and during that interaction, oxytocin is triggered optogenetically by light pulses. You can see that during the recognition phase, we have that not only were there more events of exploration between these mice, but also the amount of time spent exploring was longer. So this served to put forth another question. Could oxytocin be a booster for social interactions? Now, the effector targets for oxytocin release basically the oxytocin release would be centrated around the paraventricular nucleus of the hypothalamus. So it's a bit medial in the brain. And you could see that we have different effector targets. So where if we have oxytocin release, it might go out and affect different areas of the brain. So the oxytocin release affects primary olfactory regions, including the anterior olfactory nucleus. And the PVN, or hypothalamic oxytocin neurons, we can study their projections by looking at studies that have done viral injections that actually label from this area of the brain to its different effector targets. And we could see that, for example, we have oxytocin inputs that go to the AI, or agranular insula, so that's anterior, and also to the amygdala, for example. Now, in a second scheme of events where we have a social interaction, and we want to look at state-dependent processing, which includes arousal, exploration, we then conducted two subsequent experiments. One where we triggered oxytocin release during a social interaction and put them in the scanner. And also when we put the mouse in the scanner and we trigger the oxytocin release without a social interaction. So in the scheme where we have a social interaction, and in this case, the animal that's going to go in the scanner is receiving the pulses to trigger the oxytocin release. And also to kind of get a little visualization, this would be the paraventricular nucleus of the hypothalamus. And here we have viral labeled expression of oxytocin neurons. And we're giving those pulses for 2 seconds. And then we're measuring what's happening in the brain. So the mouse that's the interactor here, he's getting familiar with its conspecific. And we presented the odor of that familiar mouse and also an unfamiliar mouse that is completely novel. And what we wanted to do was parse the underlying activity correlated to each. And so if you do a contrast, what you can see is that when the animal is familiar, you have more activation of cholinergic areas like the diagonal band of Broca, the anterior olfactory nucleus, and the bulb. And where it's unfamiliar or perhaps more novel, you have this recruitment of the anterior insular cortex. And looking at the results from experiment 3, where we just give the laser in the scanner, what we can see is that we have, for example, some overlap. However, there is still a distinction. So what is perhaps one of the more striking distinctions is what you have ventrally. This is the ventral striatum. It's important for reward processing. You have more of that occurring if you just give the laser. However, I do want to point out that because we contrasted here and we basically looked at differential activity patterns, why we don't see the ventral striatum or reward region here is because it's actually lighting up for both. So it doesn't matter if it's a novel or a familiar social stimulus. And what's also quite interesting is that when we look at the anterior pole of the mouse brain, where we have the olfactory bulb and the anterior olfactory nucleus, you can see a nice segregation where you might have one area allocated to the processing of social stimuli, whether they're familiar or just for the oxytocin release. So this helps us get a little bit of an idea at the mesoscopic systems level, what could be the underlying signatures related to such processes. So the processing of chemosensory activity was studied here in the passive state, so no task engagement, and it was examined after perturbation of integrated circuitry, in this case with the oxytocin stimulation. The insular cortex was highlighted as a primary brain region for taste processing and is also implicated in the processing of olfactory stimuli. Differential patterns of activity across olfactory stimuli were observed at the mesoscopic level using fMRI, and optogenetic stimulation of oxytocin within the hypothalamic paraventricular nucleus was correlated with increases in both fMRI activity and reward processing hubs. Bilateral activity in the olfactory tubercle, nucleus accumbens, that's the ventral striatum, heavily implicated in reward processing. Also the amygdala and the left agranular insular cortex. Given that there was no prior social defeat between these animals, all social odors were assumed to be appetitive. However, the sniff rate is a critical parameter to help infer the appetitiveness and potential valence of stimuli. So without further ado, I'd like to thank the team. And the work that was done in mice was led in the lab of Wolfgang Kelsch, and that is in Germany, in Mannheim and Mainz. And the work specifically was done in coordination with the PhD student, David Wolf. And the work with the non-human primates was done in the lab of Henri Avard within the Department of Mycos Logothetis, and to thank also our support there. And yeah, so without further ado, I would like to thank you for your attention.